actually happening, whether there is a change of the accused material, calibration or control, or there is a change on the procedure or something happening with the instrument itself. Because we have to be cautious, it's easier to correct it at once than have to check all the specimen before that something happened.
I think it sounded to me like that was possibly due to a lot-to-lot variation. And I think if you rely on QC to detect lot-to-lot variation, you will wait a long time. I don't know, it depends on the assay, but often people will run a QC sample at the end of every shift, or once a shift or once a day. When you take into account how long it will take conventional QC if you run that against 2SD, how long it will take you to pick up that there's been a significant change, it could well be days. So if you suspect that you've got or you detect that you've got a lot-to-lot variation between reagents, you need to test before you put the new lot of reagent in. If you know that it's highly likely that there is a chance of lot-to-lot variation, you need to test each new lot using patient samples, stored patient samples, and not rely on a QC sample to pick up lot-to-lot variation. And I think what Ken said is also very true. That's one of the advantages of using patient-based QC. That the patient means or medians that you're using, you're tracking them all the time. And even if you have to use 60 samples to give you a good mean, that will be, I suspect, you will detect a shift long before conventional QC will detect that shift.
Okay, thank you. Thank you.
I'll tell you, I will tell you what we do in our laboratory, because it agrees with what's been said before. Whenever we get a new reagent, we run it in for a week. We run QC on that reagent for 1 week before we report any patients. Because as Tony says, it takes a while to realize whether that QC is truly in, like your 10x score or 41SD scores. It takes a while for you to be confident that the new lot is okay. Now, it does mean you've used up, well, it's not that much. If you run the high and low QC twice a day for a week, you've done 20 assays, and out of a 500 test kit, that's not a lot not to use up once when you've started up this new reagent. So I would, if you can manage it, run that reagent for a while before you use it. Otherwise, you'll get a surprise.
Okay, thank you very much. There are so many questions coming. I will choose some interesting and the same related questions. How would you adopt, this is from Rukman Awang Hamad, how would you adopt ISO for labs in low middle income countries? Would it be possible for them to achieve acceptable requirements? Maybe Prof. Tony Bedrick or want to answer first?
And that's a tricky question, because it depends on what resources the laboratory has in place. That's, you're talking about a wide range of capability, and in terms of there could be issues with getting reliable reagents, being able to pay for sufficient QC and EQA. But really, the 15189 represents a sort of the base level of quality that we demand in laboratories. And there's a lot of aspects of 15189 that are not just about assays. They're about training and all other sorts of procedures that need to be in laboratories in terms of ethics and maintaining equipment, etc. So all of those things should be applicable even in those resource poor countries, I would have thought. It may be that some aspects can't be controlled as well as others because you can't afford sufficient controls, but certainly I would think that laboratories everywhere should aim to get as many of the clauses of 15189 in place as possible. It really does represent the base level of the expected quality to reduce patient risk.
Dr. Shan, any experience maybe from Indonesia about adopt this ISO for labs in low and middle income countries?
I think in Indonesia, we follow the ISO 151, because the ISO 15189 requirement are the requirement that how to provide qualities like Tony said. So it should, even though there is limited resources, but the standards should, the procedure should follow in such way that the person can show that it is still well managed and can still prove that the validity of the result is obtained, such like quality control. Usually in the big lab, they do it every day. But in the small lab, they do it only once a week. They don't have to do it every day. They can do it every time they have specimen. And for the POCT, it's according to how stable is the test itself. You don't have to do it every day. Maybe if it's stable and if the equipment itself have like the internal control or something. You don't have to have the laboratory information system to have the proper, to have fulfill the management requirement. Even with the manual one, you can fulfill that. It's according to me, but because we also assess the small laboratory, just how they can manage their test according the use for the patient. Usually they use POCT. Most of the lab use POCT.
Okay, thank you. There's a question for Prof. Kensi Karis. How do we calculate, this is from Anonymous, how do we calculate measurement uncertainty for a single assay on multiple instruments? Do we calculate individually or do we combine into a single uncertainty? And second question is, should bias be included into measurement in uncertainty calculation?
It's a very good question. And it's, in a way, when you've got your SD on an instrument, you want to work out whether that instrument is changing. Now, so there you do need an SD which monitors the instrument, because you don't want the instrument. And instruments are different to each other. In our laboratory, we have many. You'd be shocked at how many instruments we have. We might have 10 instruments measuring sodium. And there is a bias between one instrument and the other, permanently different by 0.1 or 0.2 of a millimole of sodium. So we don't want to wait until one instrument, which usually goes a low bias, becomes like the instrument with a high bias, because that instrument's deteriorated. So measurement uncertainty can be used to monitor instruments, but the ultimate purpose of the uncertainty, which I described in my talk, is to interpret patient results. And if a patient can go from instrument A to instrument F, it means that uncertainty the patient sample's using is the total uncertainty of all analyzers. So we do have an uncertainty across analyzers, and that's what we give to the clinician, because we're not sure which analyzer the patient will go on. And so even if there's a bias between one analyzer and another that we're not choosing to correct, we'll include that bias in our overall uncertainty. So as long as that overall uncertainty is fit for purpose, it doesn't matter. But the overall uncertainty is what's so the short answer is the important uncertainty is the across analyzer uncertainty as far as patients are concerned. But for your quality control system, you might want to know the uncertainty of each different analyzer so that you can work out whether an analyzer is starting to deteriorate.
And in fact, I was just going to add, a broader question. I mentioned that, the different EQA audiences, there's the lab and there's the assessors, but there's also really, the people who write guidelines, or even the people who interpret our results. They really need to know what's the uncertainty generally across all laboratories. Now, it's good to know that the uncertainty from laboratory A is 2%, and maybe that the uncertainty from laboratory B is 3%. But honestly, when they interpret results, they need to know what would be the general uncertainty across any laboratory, so I can factor that into my clinical experience when I try and interpret results. And particularly, I mentioned the issue with ferritin and some other tests where there's a really tight cutoff. One question is what method was used to determine that cutoff, but also what's the general uncertainty about that cutoff, in all laboratories around your country. That's critical with the interpretation of results, both those results and also the results where you're monitoring a patient, and what's the sort of change that we can expect purely due to laboratory, uncertainty as compared with pathological, changes. So we shouldn't just be interested in what goes on in our laboratory. We need to be interested in what happens with the results and how they're interpreted. We always need to sort of look outside our little silos and say, well, we've got a great result, but how does that compare with the result from another laboratory? We've got a really tight uncertainty, but how does that compare perhaps with another laboratory's uncertainty? Because somebody else out there doesn't really care about those things. They just want to know, how can I interpret this result? What does it mean to my patient? I think we should never lose sight of that.
Okay, thank you. Because you already mentioned about the EQA, there's a question. Can we use EQA performance to replace UCAL, uncertainty calibrator, for MU calculation? Prof. Tony, maybe you would like to answer this question?
I think if you use the right material, I think you can certainly get from EQA if you have enough samples and it's material that truly reflects what's going on with your patients. I think you can use EQA to calculate MU, and in fact, it's probably even safer if the EQA provider that calculates that MU for you or for your instrument group. Really, what in terms of the bigger picture, how do people interpret results? They need to have an idea of what the MU is about that result, and it's probably better because as well as the issues about what data do I use for MU, people often don't seem to calculate it very well either. They have their own formula for calculating MU when you go around from one lab to another. So it would be good to have independent assessment of MU, and perhaps that could be given as part of the service by an EQA provider if you do enough samples and the material really is appropriate to do that calculation.
Can I ask a question of Tony, because he's a world expert on EQA?
Yeah.
The BIRAD Unity program does collect the information on the lot of reagent. So there is some information in that program, the internal QC sort of program, to be able to differentiate lots. How useful do you think it would be for EQA programs to collect the lot information on reagents rather than just the manufacturer?
I think it's critical. I never used to think it was important once because I'd been brought up as a chemist, but then I got exposed to serology and I realized how critical lot-to-lot variation is. And now I think we're discovering in chemistry as well that in fact there's significant lot-to-lot variation as well, not just in immunoassays, but also even in some of our other assays. So I think it's important. It's very difficult for laboratories to do it because we change reagents and we forget that the lots change. It has to be automated in some way. But I think the more information we can gather about our assay, with lot-to-lot, with the lot number as well as the calibration number, I think that all will add to the information that we've got about what's going on with our analyzers. And more importantly, if we can compare how our performance against somebody else, not just using the same analyzer and the same reagents, but the same reagent lot number, that's when we'll really start to learn more about what's going on and potential sources of error. Because manufacturers make mistakes. Their processes aren't not completely without error.
Thanks.
Okay, thank you. Very interesting discussion. And I think there's another question related to this also. How about for the immunoassay parameters, such as hepatitis B, HBe's AG, and anti-HIV? If our lab result shows borderline for the uncertainty, should we just refer it to the other lab that has different method? As we know that for the immunoassay parameter, especially for the infectious disease, is there any like the traceability or what? Because we know that the immunoassay parameter that have like a cutoff with the numbers of index or as per CO. Maybe Prof. Ken Sikaris would like to answer these questions.
Tony mentioned the problem of serology assays, and
Yes.
often they're the positive, negative, or borderline assays. Now, it is possible to deal with them quantitatively, but it's sort of almost artificial because what the clinician sees is the qualitative result, positive, negative, or equivocal. And so now you could repeat an equivocal result, and if you repeat it often enough, it might become negative or positive just because of the measurement uncertainty in the process. And so that's just generally considered to be an inadequate approach. The best way to, the standard approach for serology, and I'm not a microbiologist or serologist, but is usually to go to an independent assay to see whether it also gets a negative or positive result, rather than to play with this grey zone of uncertainty of borderline and try to, because generally serology assays aren't monitored truly quantitatively like a lot of our chemistry assays are. They're monitored qualitatively. So I generally, that's what we do in our laboratory. If we've got an equivocal result, we usually go to another platform to confirm whether it's likely to be positive and negative, rather than to repeat the analysis on the same platform.
How about you, Dr.
I think that's key. Sorry to interrupt. I think the other thing that's key with serology results, as with a lot of other results that are qualitative, is that there needs to be really good information given back to the clinician in terms of how to interpret this result, particularly if they're on the borderline. There could be borderline for all sorts of reasons. It could be the patient's getting better or worse, or it's too early. So I think it's really important that lots of clinical information is given to allow the clinician to interpret the result with assays such as that.
Okay, so just repeat with another method or another brand analyzer. It's not a solution to this, right?
If you've got that ability, that's the solution. But if you don't have that ability, I'm not really sure how many repeats are going to give you more confidence in a borderline result. It's borderline by definition.
Dr. Shah,
Maybe
please.
I can ask to Dr. Sikaris. Is it possible to run by two other different methods that are more sensitive or have the more precise? And
yeah, ideally the second method is better in some way. Like it's a better antibody, a better. I mean, often we're doing serology and confirming by PCR. Like that's ideally the second method is a better method. And even in drug toxicology, which is another example, our screening method is qualitative, and our confirmatory method is a better method, like chromatography. So ideally, the confirmatory method should be a better method. It doesn't really make sense to say I'm going to repeat it by a method that's worse. So it's much more logical to repeat it. If you repeat it with an equivalent method, I agree with what you're implying, was that so this method says it's borderline, this method says it's positive. Well, which one will I believe? Do I have to do another one as a tiebreaker? So it's awkward when you're repeating with equivalent methods. Ideally, you're repeating with a better method so you can have confidence in the second method.
Okay, thank you very much. And there's also another interesting questions. The result of D-dimer, I think that's also a problem for us too. I experienced variety results for the dimer sample performed using different analyzer or systems. The difference among systems were so great. So how is the development about D-dimer traceability? Maybe about traceability, Prof. Tony Patrick, I think.
I honestly can't answer that question. I just don't know. I don't know anything about D-dimer traceability, unfortunately.
Okay. Any speakers want to answer these questions? But
No, I don't.
D-dimer traceability.
One thing there is reference material for the dimer at present. And the dimer consists of different particle, the long one, the short one. So it depends on which epitope they will target. So that's we found many difference when doing the dimer with different platform.
Oh.
At present, so as far as I know, there is no reference material for the dimer.
Okay. Thank you. Maybe let's jump into the POCT. As we know that in ISO 15189 2022, there's like another rules for POCT. So there's a question from Lucia Sinchugunawan. How do I select the best POCT equipment? Should I make study comparative with the gold standard technique? Maybe this means to the analyzer. Is there any calibrator for all POCT equipment? How to calibrate POCT example for the lipid profile? For the first question, the study comparative with the gold standard technique, maybe I will ask Dr. Xian, and the calibrator of the POCT, maybe I would like to ask to the Prof. Tony Patrick. Dr. Xian first.
I
Okay.
think it depends on what is the intended use of the POCT test. Because for screening, for diagnosis, or for monitoring, the intended use will be different. And for the POCT, if it's used by the different operator than the laboratory technician, then maybe we should consider also the easiness of use and also the maintenance and also the stability of the reagent itself. So not just the specification of quality, but also how to use it, because it is difficult to keep the competency of the person outside the laboratory, because they just do it once in a while, not every day, something like that for the POCT. And I think what's the other question, Dr. Tirsa, is
About the calibrator,
standard?
For the POCT equipment and how to calibrate for the lipid profile of the POCT.
If the POCT is in an institution like a hospital, then maybe it should be compared to the method in the central lab. But some of the laboratory have it POCT, just POCT and do it in the clinic, something like that, but not doing it in the laboratory. Then still they have to have the EQA for comparability and the internal quality control to ensure the monitoring of the quality.
Prof. Tony, you would like to add about the calibrator, maybe?
Yeah, I think that's fair enough. I always think that QC and what Dr. Shan said about fit for purpose. That's the whole crux of the matter with point of care. It's often used for a different purpose, but still you still want to maintain comparable results with other systems. But remember, with point of care, usually point of care you're using a finger prick, so it's a different matrix. It's whole blood. There's going to be different values, I always think that there are two aspects to point of care. There's the instrument itself, so that's there's usually a cartridge or some device like that that's used, and they're really mass produced in a factory, and they've got really very high tolerance, so there's very little that can go wrong. But QC of batches is important, just to make sure when a new batch of cartridges or whatever comes in, that you do some sort of QC to ensure that you're controlling that the cartridges are giving the same result as they should. But more importantly, with point of care devices, I think is the operator. Most errors are related to operator error, not just in the process of putting the sample in, but also using expired cartridges, recording the result, ensuring that the result goes somewhere, goes into the patient record, maintains the equipment, maintains their own training. I think that operator competence is the key to the issues with point of care devices. To be honest, that's where the focus should be.
Okay, thank you. There's another question about POCT also from Dr. Rosinta Christina Solin. The new standard requires that the frequency of POCT QC be based on stability and risk. Does this have guidance from ISO? How should we evaluate this stability? Thank you. Maybe this is for Dr. Xian first.
Okay. I think there is some journal that explaining how to do the IQC in primary healthcare that consider the risk of the test itself, and also the easy the user friendly of the method itself, and what kind of instrument is it itself, whether it's a cartridge based, manual based, or liquid based, something like that. And also with and also they consider also the frequency of how many tests is done by the operator, and then they decide how many times should the IQC be done. I think the stability can be seen from when they are doing the IQC day by day. They can see whether the tests are stable or not. And also they can see when they watch the patient result, whether the result from the test are stable or not. That's one of the thing they can study to see the stability of the POCT test.
Okay, thank you. There's a question from Julie Shervan to Prof. Bedrick. If the lab has demonstrated that a method is not traceable back to the reference standard material, it means by running patient sample against the manufacturer's calibrated versus the SRM, rather than applying a post-analytical correction factor, would advise that the lab use the SRM value to recalibrate the method in addition to informing the manufacturer?
Labs don't usually have those materials. Those standard reference materials are usually expensive, and so it's not that easy to get that material. I guess if you've got that material and you want to recalibrate your assay so that you're sure that it's traceable, that would be one option. But I think laboratories always have to be careful about doing things like that. As I said earlier, the problems we have is related to traceability are that different assays get different results on the same sample, and the issue with interpretation comes about how are these results interpreted? Are they interpreted against a reference interval, or are they interpreted against a decision point, or are they interpreted in terms of looking at changes in a patient? If a lab has a reference material and they calibrate their assay and therefore change, relatively speaking, their results compared with what they were getting before. They need to be aware that they haven't changed any of those other factors. You know, is it still valid to use the decision points that were being used before, the reference intervals? What about patients that have already been, results have gone out and they're being monitored? I think that there are lots of issues that you need to be aware of. And if the whole assay has changed, you may, a factor would be, I think, important to bring the results back into line with what you were getting before. But I think to bring in an external standard, if you've got it, I think that you'd have to think about that very carefully.
Okay, thank you. There's a question to Prof. Ken Sikaris. Should the uncertainty value be evaluated at the clinical decision limit rather than the reference value? What do you think?
Very good question. Because the measurement, that ultimate purpose of measurement uncertainty is to fulfill its clinical purpose. The clinical purpose is most important at those clinical decision points. So now sometimes we don't have clinical decision points, like for sodium, there's 135 for the lower limit and the upper limit. So it's important to know the uncertainty at the lower and upper reference limit. But for other things like glucose, which I showed, or A1C, it's more important to know the uncertainty at that clinical decision point so we can decide whether the patient is clearly above or clearly below the decision point. There are some other decision points that we use for all assays, like the critical limit. Like, so we'll phone through a potassium when it's over 6.5, let's say. Are we confident that it's above 6.5? Does our uncertainty estimated at 5.5 apply to 6.5? So it is handy to know that, and usually the manufacturer or laboratory has an idea of the functional performance of their assay. What is the range of reportable results, the analytical range? So I think that the answer is yes. The best place to do measurement uncertainty is at the clinical decision points, and the fallback is to understand how your assay varies in its precision across the analytical range.
Okay, thank you. There's another question for Prof. Tony Bedrick from Julie Sharvan. For a measurement with two reporting units, such as percent in A1C and also SI units in A1C, is it possible for one unit to be traceable and not for other?
I think that's an interesting question, but really the units are transformable. You can transform one into the other. So I think therefore. And they are traceable, so I think they're both traceable, but you just transform one into the other.
Okay, thank you. Question for Dr. Xian from Chong Chan Eng. For CE-IVD diagnostic products that have been not approved by local regulatory authority to use in diagnosis, but imported as RUO, research use only, should the lab performs a full validation or verification only?
Okay. First is you can ask from the manufacturer whether they have already done the validation study. If you are not sure whether the validation study has been performed, but I think the local regulation should be followed first, because the regulation is in each country will be different. Either this is an RUO or not. But if the manufacturer say that this is already validated, maybe you have to make ensure and make them give the validation study and see whether the validation study is already appropriate or not. Because usually it will be the IVD will be regulated as IVD also, but maybe something else that make it imported as RUO, maybe because different intended use or something. This must be clear first. Maybe Dr. Tony or Dr. Ken have any experience regarding this?
I think with limited experience in assays like immunohistochemistry, with these sort of research reagents, I think you need to do local validation.
Okay, with the local validation. How about you, Dr. Sikaris?
I mean, we have regulations in Australia, and generally when you've developed an in-house assay, you're allowed to use it, but you should warn the clinicians that this assay is unvalidated. So, and do whatever validation you can. Ideally, send that validation to the national authority so that they can improve the validation of your assay. But if you don't, and you still want to use it for research or for clinical purpose, you have to be very clear to the users of that the lack of external validation of your assay and the uncertainty, that any uncertainties you have in your results, because I know that, you know, I mean, we do want medicine to progress. We want new assays to be developed, and they have to start somewhere. But the important thing is to be transmitting, you know, the confidence you have in that result honestly to the clinician who might be using it for the care of the patient.
Okay, thank you. There's a question from Zulhainan Hamza. The alignment of ISO 15189-2022 with other ISO standards such as 17043, 17025, and others will strengthen the implementation of the ISO 15189. But how far that we can apply those other ISOs as only a guidance for the laboratory or as part of a needed document during the assessment? Thank you. Maybe this is for Dr. Xian.
Okay. As we can see when we read the ISO requirement, it's something like general, not step by step how to implement that requirement. So the guidance give the step by step or more details process on how to implement the requirement. And this is one of the things that should be followed by the lab when they implement ISO 15189. But other standards, like the national standard, national regulation, also apply in the implementation of ISO 15189. So it's not only just for presenting it as having it as a guideline and not implementing it, because they said that they or we just have it as documents, if I'm not mistaken, the question.
Okay.
So you have to implement the standard.
Thank you. Thank you very much, Dr. Xian. And there's a question to Prof. Tony Bedrick, anonymous. How to set a reference interval for each parameter and each age classification? Is it okay to use reference that is given from the manufacturer or maybe from the studies, the journal?
I think reference intervals are key to interpretation of results. I think you can use the manufacturers initially their guidelines, but really the best option is to adopt a reference interval based on a lot more patients than you've got no idea when a manufacturer gives you the reference interval, how much data they used, how many patients, etc. What was the range of patients that they used? You know, I think that laboratories, or better still, national organisations, where possible, should develop reference intervals for general use across laboratories using really patient samples. I think the indirect method is the more effective way of developing reference intervals, and labs should not just adopt, unless it's a very unusual assay, very rare assay, they shouldn't adopt reference intervals from manufacturers. But I might ask Ken, who's the expert on reference intervals, to give some more input into this question.
Please do.
Now, the only thing I'd add, I'm on a committee called the IFCC Task Force on Global Reference Interval Database. So they're aiming to provide two things: to provide a database so that you can go and hopefully, let's say you don't trust the manufacturer's reference intervals for albumin. Has anyone ever published for Malaysia or for Singapore or in Indonesia? Has it been published? We don't have a way of easily finding that information. So one of the things we're hoping to do is provide a website where you can find that information, like the biological variability database that EFLM have had. And the other thing which Tony said was that I think laboratories underestimate how useful the data that they have is in determining their own reference interval by this indirect. When you pull out all of the data for your albumins in Indonesia, it'll follow a Gaussian distribution and it'll reflect what the true reference interval is. You just need the statistical tools to be able to understand your laboratory data. And it doesn't even have to be an individual laboratory. You can pool, if you've got the same method in different laboratories, pool the data from different laboratories, because you do need a fair bit of data to do this. So I do agree with Tony. This is the way of the future, that we don't rely on manufacturers testing 40 German people 40 years ago to tell us what normal is. We really need modern methods of determining reference intervals. I will say that I wouldn't ignore the manufacturer's kit insert. Some people use a textbook which is 40 years old rather than a manufacturer's kit insert which is 10 years old. Well, that's a mistake too. So really it's to use the most up-to-date information that applies to your method and your patients. And if that's in the kit insert, well, then that might be the best interval you can get at this stage.
Dr. Ken, usually in our country, we have to comply to the regulation of the ISO that we verify the manufacturer reference by testing 20 healthy individual. But the difficulty is it cannot cover all the age
partitions. Exactly. No, it's very hard.
The reference interval.
Look, in the standard, it actually says that you don't have to do everything, unless you think they might be different. If you think that thyroid behavior, the thyroid, the TSH molecule is different in pregnancy than in others, you might have to validate pregnancy separately. But if you can assume that the analyte is the same, or the matrix is the same in all partitions, then you only have to validate one of the partitions. It says that in the ISO standard. So you don't, I agree with you, ideally you should be, but it's very hard. I mean, when you consider all of childhood, neonates, infants, puberty, young adults, postmenopause, premenopause, pregnancy, first trimester, second trimester, it's impossible for any laboratory to validate reference intervals. But you should at least validate one, and ideally the one which applies to the most common partition in your testing population.
And I think it's also a little bit difficult for the pediatric reference interval.
Yes.
It's quite challenging.
Yeah, well, the pediatric thing is, the problem is that pediatrics don't, even when I said neonate and infant, it's not as if when they're 1 month old, they suddenly change overnight to be a different type of child. It's a continuous change. And so the way forward on that, and Jacob Zierk, who's a young German pediatrician who's on a IFCC committee, he's really trying to promote the use of continuous reference intervals. Now, continuous reference intervals are very difficult for laboratories to adopt because our LASs, our reporting systems, don't allow usually for continuous reference intervals. But we can still provide resources like a growth chart, a piece of paper that says this is how the reference interval changes in a child, or in pregnancy or anything else. So you don't have to provide the information on a report. You can just say to the clinician, we've got that information available if you want it.
Okay, thank you very much. There's a question from Busadi Pratumfinit. Sorry if I mispronounce. The question is to Prof. Sikaris. From the graph compared between CV of EQA and CV of IQC, CV of EQA means the CV from the participant lab or CV from repeated EQA samples.
Well, yeah, it's sort of touched on what Tony was saying. So the CV in that graph was the CV for that laboratory. So for the samples that that laboratory submitted to the EQA project. Now, it doesn't mean that was the CV for the EQA sample, which we repeated 30 times in the laboratory. That's not what it meant, that graph. But that would be, I don't see what the point of that is, because you may as well repeat a patient or your internal QC sample if you want to get that sort of estimate. But I agree with Tony that the overall CV contains some new information. Like if you compared your internal QC with the CV of all laboratories, that's a different question altogether. But anyway, that graph was just the results that individual laboratory submitted to the EQA program and how that QC compared with their internal QC.
Okay, thank you. Let me see, let me check first if there's another. Oh, this is about the POCT. Are there any recommended proficiency testing for POCT tests? Can split samples be used for POCT tests? Maybe Dr. Xian would like to answer these questions.
Actually, what means by POCT is not only manual tests or rapid tests or the very simple tests. So it can be also the big equipment, but put outside the lab. So there are many proficiency provider that can handle. There are many proficiency provider for the POCTs. You can have the blood and glass analyzer in the ICU or hematology analyzer in the ICU, something like that. There are proficiency test provider for that. But how to choose it is better to have a proficiency provider that is already accredited with ISO 17034, if I'm not mistaken. Then it means you don't have to obtain data from the provider for the appropriateness as proficiency provider. So choose the one that already accredited, but 170 or 0.43 ISO. And can be we can do the proficiency testing for VOCT by doing one sample and comparing it to other labs, but not to the same lab, in the same lab. So we know compared to it was compared to different labs.
Okay. Okay. Thank you very much, because we are running out of time. I'm very sorry if there are questions that I cannot read or ask to the presenter. If you still have questions, maybe you can email us and we will ask your question to the presenter. And I would like to remind to all the participants, for the attendee link absent, it will be given directly to the email. So if you need, please allow me to speak in Bahasa, because many Indonesian participants will take the CME credits. Jadi untuk para peserta yang untuk pengumpulan SKP di Pelataran Sehat akan dibuka hari ini pukul 5 sore untuk mengikuti pelajaran. Dan untuk link absen itu tidak akan diberikan melalui Zoom ini, tetapi secara otomatis untuk seluruh peserta yang mengikuti Zoom ini itu sudah terdata oleh panitia dan akan diberikan email untuk kuesionernya. Jadi harap tolong diisi tentang kuesioner dari acara ini untuk bisa mendapatkan sertifikat dan diverifikasi di LMS Camcast. Jika tidak mengembalikan kuesioner, maka tidak akan diberikan untuk akses pembelajaran di LMS Camcast. Seperti itu. But for the international participant, all the registered participant we will give the certificates of all the attendants that who already joined this Zoom webinar. Once again, I would like to thank you for all the 3 speakers that have marvelous presentation. It's a very clear, very interesting topic. Maybe I would like to on cam all to take a picture of all the speakers. Who will take the pictures? Pak JP, maybe you would like.
Okay, 1, 2, 3. Nice. Once again. Okay, thank you.
Oke, terima kasih untuk semua partisipan.
Dr. Sikaris.
Thank you very much, Prof.
Thank you.
Patrick. Thank you very much, Prof. Ken Sikaris. Thank you very much, Dr. Shan. Our session today is finished, and I would like to remind to all participants that we are going to have another APFCB IACC webinar. Can you please display the event, Pak Cepi, for the next session? Yeah. For the next session will be held on the July 27th. It's about effective of IQC planning, Sigma, and troubleshooting of IQC. September 28th, about external quality assessment, how to select the EQA, harmonization of the EQA, the interpretation and troubleshooting of EQA. And November 23rd, will be method validation versus method verification, steps of method verification, reference interval, and decision limit. I think maybe there are questions that still not answered today about the method validation, method verification. You can join us on another APFCB IACC webinar 2024 series that is supported by SNIPE. Okay, thank you everyone. Have a good day. Bye. See you next time. Pak Cepi, jangan di-end dulu ya Pak. Mau di copy paste
Baik, Dok.
pertanyaannya.
Izin live ya, Tir. Sukses ya. Puji Tuhan beres semua.
Ya.
Keren, keren.
Yang di YouTube tadi nggak ada pertanyaan ya. Aku kayaknya tadi belum sempat ngeliat-ngeliat di grup. Sepertinya nggak ada ya.
Tidak ada, Dok, terkait susah.
Oke, ini tadi saya udah beberapa sudah saya catat sih pertanyaan-pertanyaan. Apa namanya? Saya copas bentar. Ini tadi saya sampai mana ya?
Q&A bukannya tersimpan ya, Kang?
Hah? Aku nggak tahu nih. Simpan.
Harusnya bisa ketarik itu.
Seingat saya bisa ketarik sih. Saya sudah agak
Bisa
lupa
ketarik.
juga megang. Sudah lama nggak megang Zoom.
Oh gitu.
Tapi dokter mau ini boleh dok di kopas-kopas dulu aja, just in case kan dok?
Iya, supaya buat ini kan, apa namanya? Supaya buat apa? Tapi ada beberapa yang nggak saya ini kan, soalnya ada yang berulang sama ada yang apa namanya? Ada yang ini. Ini yang di YouTube udah di off-in kan ya? Kita ngobrol nanti
Sudah.
kedengeran di YouTube.
Ini kalau nggak di-share, calling nggak akan jalan nih.
Oh gitu. Tunggu ya Pak, bentar ya Pak. Ini udah tinggal dikit kok. Tadi saya udah
Baik,
sambil
Dok.
itu udah beberapa yang udah saya ini sih.